Hello, and welcome to today's webinar, Cryopreserved Autologous CAR T Workflow Processing. I'm your host, James Crosspayne with Cell and Gene. I'm joined today by two industry experts, Robert Bilski, Field Application Scientist with CAR Biosystems, and Ali Reza Abazari, Senior Director of Cell Processing with BioLife Solutions. I'd like to thank you, the audience, for attending today's event. Throughout today's session, please share your thoughts with us using the Q&A box on your screen. We will answer as many questions as possible, and any we miss, I'll pass along for follow-up after the event. This question box can connect you with me if you run into any trouble, but please try refreshing your browser first, as it will fix most common issues. Our presenters have also asked you a few questions that you'll find on your screen. Please take a moment to share your experiences with us. Finally, today's event will be available on demand. You'll receive an email with a shareable link shortly after we conclude. Now, without further ado, let's begin today's presentation. Welcome, Robert.
Thank you, James. So as James said, hello, everyone. My name is Robert Bilski. I'm one of the field application scientists here at CAR Biosystems. And I'd also like to introduce my co-host, Ali Reza.
Hi, everyone. My name is Ali Reza Abazari. I'm a Senior Director of Cell Processing and Biopreservation at BioLife Solutions, and happy and excited to be here to share our data and findings with all of you today.
Thanks, Ali Reza. So today we'll be speaking on the cryopreserved autologous CAR T workflow and how leukapheresis product, also known as Leukapheresis processing, can be streamlined across concentration, wash, and media exchange for cryopreservation. So setting the agenda for today's webinar, we'll cover a variety of multitudes, such as traditional Leukapheresis workflow, introducing tubular bowl centrifugation for the Leukapheresis workflow and other cell therapy workflows, highlighting data looking at Leukapheresis washing, concentration, and harvest. I will then turn it over to Ali Reza to discuss cryopreservation and thawing, as well as the impacts on downstream CAR T manufacturing. We'll wrap up by discussing some ongoing studies for this project, as well as call out some key takeaways from the data collected thus far. So let's start by looking at the traditional leukapheresis product processing workflow. So kind of taking a step back and looking at the initial data or the initial Leukapheresis that's collected through apheresis, there tends to be a variety of process steps that have to unfold prior to getting to the final formulated product. And that starts with traditional platelet washing or debris removal, going into, from there, a variety of process steps such as cryopreservation, expansion, thawing, cell selection, gene editing, or other avenues going into that final formulation product. So CAR Biosystems and BioLife Solutions all provide solutions throughout the entire cell therapy workflow, which will assist the developer from start to finish with achieving their final critical quality attributes going into that formulation. So for this collaboration, we've been working through a variety of donors and variety of testing conditions to see whether or not we can take this apheresis product going into final formulation step. But for the next few minutes, I'm going to be discussing the washing and concentrating step of the Leukapheresis washing, which leads to cryopreservation and showcasing metrics such as viable cell recovery, viability maintenance, cell population ratios, as well as washing efficiencies. So kind of transitioning into some challenges with Leukapheresis processing in particular. There's a multitude of variables to be considered, such as starting material variability. So with each donor from a Leukapheresis processing tends to have different cell populations, different debris populations. So having the ability to overcome some of those variabilities from donor A to donor B to donor C can really lead to an impact on overall further downstream processing. We also have operator variability. So each operator, when collecting the Leukapheresis or doing that cell therapy workflow, tends to have that potential manipulation of manipulating that Leukapheresis differently, which in turn can impact overall process performance of cells further downstream from that initial Leukapheresis collection and washing. We also have consistent cell recovery issues. So because we have those variabilities associated with starting material and operators, that variability in result can lead to, filling challenges, formulating challenges, and further expansion if these poor cell recoveries are repeatedly discovered and occurring throughout the process. Traditional technologies for Leukapheresis processing tend to be utilizing open separation technologies. So being able to mitigate that challenge associated with open processing from a sterility component or a contamination component can really play a factor as well in further development. And then we also have cell washing concerns. As cell washing is critical for platelet removal and other debris removal from that initial Leukapack, being able to effectively wash in a fast processing time while effectively removing debris is really critical for this initial Leukapack washing. So how can some of these challenges be addressed? And with that, I wanted to introduce single-use tubular bowl centrifugation, or the unifuge, which can be utilized for help combating most of these challenges. So whether at that initial Leukapack harvest or all the way into that final cell harvest or for pre-formulation step, the unifuge enables reliable but also scalable and simple processing of cells. So taking a look at the unifuge equipment, the unifuge yields the same user interface across the family with associated straightforward controls, in turn enabling effective and scalable processing. Within the unifuge component itself is the single-use assembly, which allows all connections to be made in a sterile and integral manner, which in turn is enabling aseptic connections for upstream and downstream process steps. Another key component for developers and end users, particularly in the autologous space, focuses on that automated production, which with the unifuge that can be utilized through its recipe-driven, push-button, and near-walkaway operation with GMP traceability for data monitoring. So looking at how the unifuge works, the cell suspension will be fed in through the inlet port on the top of the bowl as such, feeds in while the bowl is continuously spinning. So you'll see in the second image on that screen there, as the cells continue to pellet along the bowl wall, the supernatant or waste in this case would get discharged out through that, through the outlet port on the side of the machine as such. If the developer or user is looking to do any washing or media exchanging with secondary buffer, that buffer is fed in through that blue line as such through the same inlet port, allowing the cell pellet to be mixed and resuspended prior to discharging out at the very end. The very end, that bowl stops spinning, and you can end up harvesting your cells for further downstream operations in the final elution media that you were hoping to desire it to be discharged in. So kind of moving into the first part of the study, we'll cover leukapheresis, product washing, concentrating, and harvest, while focusing on a variety of data points, such as viable cell recovery, viability maintenance, population ratios, and washing. So throughout the workflow for this study, the Leukapack was initially collected through apheresis. And again, throughout this collaboration, at the time of this data, we've had 4 donors that we've been utilizing for this data to be analyzed. So these donors were collected through apheresis. That associated Leukapack was then washed within the unifuge for evaluation in comparison to manual centrifugation in this one sense. And some of the items we were evaluating were automated operation, closed system processing, as well as reproducibility. The cells recovered from the unifuge at that point were then formulated offline and filled using BioLife Solutions Signata CT5 system, where that was also evaluated for filling, cryopreservation, reproducibility as well. And from there, those cells were then taken into cryopreservation for freezing and thawing. So taking a look at the first few sets of conditions where we went ahead and took that Leukapack, fed it into the unifuge for washing, we ended up doing 7 initial tests where the Leukapack washing was done in plasma light rather than going into cryopreservation media. And upon these 7 testing conditions, looking at that initial viable cell recovery, we were able to see consistent viable cell recoveries exceeding 85% for each run. And because we saw these reproducible results across these 7 different test conditions, we ended up taking that a step further and said, well, let's try eluting the cells in cryopreservation media, in this case, Cryostore base and Cryostore with 5% DMSO. And throughout those washing studies, we ended up determining that the viable cell recoveries with Cryostore base as well as with Cryostore 5 yielded similar, if not better, viable cell recoveries, which in turn was showcasing consistent viable cell recoveries across multiple donors as well as with multiple wash medias. So what does that necessarily mean, though, if we have high viable cell recovery, but viability maintenance isn't occurring? So we took those same 7 sets of testing conditions where we washed with plasma light and eluted in plasma light and wanted to see how viability maintenance was from start to finish with the Leukapack going into the unifuge. And within each of those 7 runs, it was noted that viability maintenance exceeded 90% in each of those 7 initial testing, which really indicated very minimal impact to the cells going in and out of the unifuge. We also wanted to look further as well and see, okay, was there an impact to the cells as we went ahead and washed with the Cryostore, be it Cryostore base or Cryostore 5. And we once again saw viability consistently exceeded that 90% threshold with the different wash medias as well, showing that even if we're having different donors, different wash medias, we can have high viable cells, and with high viable cells, as well as maintaining very minimal impact from start to finish within the process. Now, we know we can recover cells with high recoveries. We know we can maintain viability from start to finish, but what does that necessarily mean if we're not recovering the right cells? So we wanted to do a comparison study to see if we were recovering the targeted cells throughout the unifuge evaluation. So 2 tests were performed with the UF Mini, and 1 test was performed with the control, in this case manual centrifugation, to see the quality of the cells being recovered. And as you can see here on the screen, the 2 bar graphs on the left-hand side were these were all with the same donor, with the unifuge, 2 test conditions with the unifuge, 1 with manual centrifugation. And across the board between the 2 UF Mini runs and the manual run, we saw nearly identical viable cell recoveries of each of the cell types observed, such as NK cells, B cells, monocytes, CD8, and CD4 T cells. So depending on the individual process, where the target cells might be NK, T, or other immune cells, and the associated goals of the developer, you will be able to achieve consistent target cell populations within the unifuge system. So showcasing to us that, with the UF Mini in general, we were able to potentially exceed, if not outperform, or to match, if not outperform, manual centrifugation in both runs, really highlighting that closed system processing, fast processing, and consistent performance from beginning of that Leukopak harvest or washing all the way to that end stage of pre-cryopreservation. So transitioning slightly to washing out and making sure that we were having effective platelet removal. Through the conditions tested, we estimated that with the unifuge, we wanted to see how does washing look in terms of effective processing time and making sure that we're able to get greater than 90% washout. So with these test conditions, we were able to see that within 1 bowl volume washing within the unifuge, we were able to achieve greater than a 90+% washout. In comparison to traditional manual centrifugation, where we see processing time for washing takes significantly longer due to the manual manipulation, aspiration, resuspending of pellet, and respinning that down X amount of times to get the desired washout. With this process with the unifuge, we can get close to that 100% washing efficiency in less than 15 minutes for processing time, not really adding a significant amount of time to the processing workflow. And because we're able to do that with the unifuge in comparison to the manual technology, we can show that effective contaminant removal in significantly faster durations of time in comparison to other technologies. So with that, I'm going to transition this to Ali Reza, who can speak more on how the cells acted post-cryopreservation and post-thawing to see true cell performance.
Thank you, Bobby. So as Bobby mentioned, one of the main goals of this collaboration between BioLife Solutions and Cryostorage was to simulate an end-to-end CAR-T manufacturing process. In doing so, we use our own tools and media and reagents that we offer. And for the rest of the process that we do not have off-the-shelf solutions. We use what is generally used and commonly used by other process scientists. For example, for cell expansion piece, we use the GREX platform. For cell selection, we use the Miltenyi platform. For cell expansion media, we used ex vivo plus ICSR. And for cytokines, we used those from Akron. So combination of all of these together with our tools, we try to simulate this end-to-end process and understand the difference that it makes if you start the process off of a fresh Leukopak or a fresh apheresis product versus a frozen apheresis product. In terms of our tools and services, as Bobby mentioned, for most of the process where we had a closed wash and reformulation, we used the CAR Biosystems UF Mini platform for cryopreservation anywhere in the process, including the upfront Leukopak or the back end of the process on the quote-unquote final drug product. We used Cryostorage CS10 that was added to the cells after wash in plasma light HSA. Then that final formulation on both ends of the process was placed on Signata CT5, which is our own fill finish instruments. That's the instrument you see in this picture on the right. And we used our CellSeal platform, which includes the 2 ml and 5 ml vials, as well as the CryoCase, which is a 70 ml high volume CellSeal container. These can be used in a closed fashion. They can be filled and sealed in a closed fashion. And then for every arm of the study that we formulated and cryopreserved, we also filled a portion of that in standard 1 ml vials or open cap to basically have a comparison with the standard practice of using the open process in 1 ml vials. So as I mentioned, the goal was to compare starting a process from a frozen material versus a fresh material. In terms of benefits of cryopreservation, we all agree. Cryopreservation essentially buys you time. It freezes the biological clock. It stops the biological clock by freezing. So that it gives you time to manage logistics, manage scheduling, facility utilization, operations, everything like that. And particularly in the case of allogenic products, using available starting material cryopreserved when needed, as needed. So it's all about utilization, scheduling, and flexibility. So we know all the advantages of that. The disadvantage that some groups may argue or have concern about is whether the cryopreservation itself has a significant impact on the process, on the product that comes out at the end of it. And so what we did with in this collaboration with our colleagues at CAR Biosystems was to get these cells, put them through two different arms of the study with fresh and frozen, and then assess the process with these evaluation areas, looking at viability and viable cell recovery post-thaw, looking at target cell populations on the front end and the back end after wash, after cryo for the final drug product for the Leukopak. Looking at transduction efficiency, we did a transduction with GFP lentivirus. We're looking at the expansion yields, the phenotype of the cells, and then also we did some additional work on that final drug product, quote-unquote final drug product, critical quality attributes that included viability and recovery post-thaw, plus how the cells functionally recovered in terms of continuing to doubling where they left off in culture after thaw. And then we looked at the phenotype of the cells after thaw and that final drug product. As I said, the two arms of the study was the fresh apheresis product comes into the lab anywhere between 16 to 24 hours after collection. And we had two arms of the study. One correction I need to make to this flow diagram is that the platelet wash with UF Mini was on the cryo arm, and the fresh arm was actually went through the centrifugation process. So a portion of the apheresis product was collected every time, went through centrifugation, platelet removal, or platelet wash, and then reformulation into the selection buffer, which then went on the Miltenyi magnetic bead columns and using Miltenyi beads for cell selection. At that point, the selected cells were then seeded for activation and transduction into 6-well plates. By the end of day 3, we transferred those transduced cells into the expansion platform in G-Rex format. We selected G-Rex because of its ease of use and the ability to reliably scale up or scale down. either in a 6M platform or a 100M platform. And then on day 9 of the process, that's one thing that we kept consistent. All of the process steps were kept consistent with different donors and different arms of the study, 3 days for activation transduction, and then day 9 of the expansion. And then at the end of day 9, we harvested and formulated that final product into the Cryostore, washed with Plasmolite, and then added Cryostore, and then went on fill finish on CT5 and our cell seal platforms. And then we did further cryopreservation and thaw on that one for assessment of that final product. Anywhere there is a star in this workflow is where we did the characterization of the product. We used the Chemomatic XM40 platform for viability and count. For the AIF product that contains multiple different cell types, we used the white blood cell count protocol. And then once the cell selection was done and we were just working with mostly pure population of T cells, we used the standard viability and count platform afterwards, protocol. And for further characterization of the cells for apoptosis, cell type, phenotype, and other expressions, we used flow cytometry throughout the process. So looking at the first step after wash and formulation, going through cryopreservation and thaw, the first set of data coming with viable cell recoveries, comparing the pre-cryo and post-thaw. Pre-cryo are the bars in pre-cryo data are the bars in navy blue, and post-thaw data are in cyan green bars, and you can compare. We did 5 different tests. So we have data from 5 different tests at this point. We have additional data that is coming. We are generating, we are continuing to generate more data on this using additional leukopaks and some of the arms of the study that have been cryopreserved but haven't been thawed yet. So there's more data coming on this. We have data from 3 leukopaks in these 5 tests. And the wash type in all of this data was Plasmolite. Plasmolite meaning that the cells were platelet washed with Plasmolite HSA, and then they were eluted in Plasmolite HSA into a final bag. And then at that point, Cryostore 10 was added to that bag in a closed fashion using our Cryostore 1-liter bag that was aseptically connected, sterile connected, welded into the formulation bag, and 1-to-1 weight-based addition of CS10, and then they were filled and cryopreserved. And so post-thaw, what you can see is in 4 out of 5 tests, we have really good, highly reproducible, viable cell recovery of the total cells in the leukopak. In one of the tests, we had some significant drop in the number of viable recovered cells in that test 4. And that was, we believe that was an issue with the thawing device that might have caused an impact on the recovery of the cells post-thaw. And so we are investigating that one further. And we're having additional data from other donors that are coming to add to this. Looking at whether we wash, whether we elute in Plasmolite, or can we do elute in something else that is more compatible with Cryostore formulation, which is Cryostore base. You can compare the 3rd set of bars with the 2nd set of bars. Very, very consistent recoveries in terms of viable, total viable cell recoveries, almost identical. That's post-thaw. We also thought to eliminate that post-thaw 1-to-1 addition of CS10 in a manual fashion. Even though it's closed, it's still manual because the bag is filled with Plasmolite, and can do a cell count at that point, and then add Cryostore 10 at a 1-to-1 ratio. Because we have to add that at 1-to-1 ratio, it doesn't matter what the cell density is. We have to add that at 1-to-1 ratio to get a 5% DMSO. And we thought, why don't we eliminate that manual step by just doing elution in CS5? And so we tested that in this case and wanted to see if there was an impact on the recovery, viable cell recoveries in this case. There has been concern, some people have raised concern that Cryostore being a viscous buffer might have an issue, might have an impact on, the shear might have an impact on viability and recovery of the cells. And using the Cryosystems platform for this purpose, we basically see no impact. So we could actually eliminate a manual step after Leukopak wash by directly eluting in CS5 on this platform. And the results are basically identical after thaw compared to pre-cryo. These are viable recoveries. If you look at the viabilities, same story, very minimal impact to viability. Even with the number 4 tests that we had some loss of the cells, that does not necessarily mean we lost viability. Viability was maintained really high. We just had some loss of the cells, which, as I mentioned, might have been attributed to bad thaw and then some clump formation that might have trapped some of the cells, but the cells that were recovered were all had high viability. Basically, if this is amazing. If you look at this, looking at these bars, the viability pre and post cryo are identical. One disclaimer is that we have done some work internally in our lab at BioLife Solutions to optimize that process. And so this is not, plug and play here. There is a bit of optimization and process development that you might also need to do on your end to basically get to this level of viability and viable cell recovery for the Leukapaks. And we'll be happy to discuss further, after this webinar. Reach out to me and we can have a conversation. So as mentioned, viability maintenance was larger than 90% post-thaw with UF mini washing and formulation and cryopreservation in the CS10 platform and CryoCase containers. So that's the total number of viable cells and viabilities. What happens to the distribution of the cells in this case? We have the data from 3 of the donors. We have data from all donors. We haven't necessarily analyzed everything. As I said, there's more data that is coming, and I'm hoping to have additional data added to this data set as we collect more data from the other donors. The data that we currently have from donors 2, 3, and 4 are collected at comparing initial apheresis product with the cell distribution after platelet wash on UF mini. We did also sample for post-thaw cell distribution for donor 4. And then once we thawed and washed the cells after DMSO wash, basically, and reformulation into the selection buffer, we also sampled that for cell composition. One thing that is striking to note is, as I mentioned, the Leukopak arrived in our lab within 16 to 24, maybe a slightly longer. And even though it was a short period of time, we had significant portion of the monocytes coming already NXN positive, which indicates they are being apoptotic. So that was interesting. That wasn't the case for most other cell types. B cells were the next group of cells that had a larger portion of apoptotic cells in that initial AIF. But when they were processed, you can see that from initial AIF to the post-DMSO wash, the monocytes are the group of cells that are disappearing and are washed out, presumably because they were NXN positive, they were on their way out, dying. And so throughout this process of washing and reformulation and thawing, they were washed out. The composition of the cells obviously shifts. Doesn't necessarily mean that we had more T cells. It's just that we had similar number of cells at a higher percentage of in that final product. Basically, for the CAR-T from a CAR-T manufacturing process, again, a disclaimer here is that we optimize this process targeting to optimize the T cell recovery out of the freeze and thaw process. If you note, there might be an impact on NK or B cells. And if the target is to recover NK and B cells from the frozen Leukopak, there might be more tweaks and optimization that is needed to basically turn the dial to optimize that for the NK and B cells versus T cells. Happy to chat further with any interested parties afterwards. So after thaw, as I mentioned, what we did was to reformulate these cells into the selection buffer and then push them through the incubation with beads for the CD4 and CD8 cells, pushing them through the magnetic bead columns from Miltenyi. The isolated cells were then used to seed for activation and transduction steps in a 6-well plate. We did this in 2 different ways. Either it was a pull down of CD4 cells, CD4 and 8 cells together. We also did sequential selection so that we can have a control on the ratio of CD4 CD8s at the time of seeding. On day 1, the transduction step was done using a GFP lentivirus. The purpose of this was to assess whether the integration, the uptake and integration of lentivirus was affected by the cryopreservation method, by the wash method, and by the cryopreservation itself versus the fresh product. Would we have the same level of transduction in the cryopreserved cells versus fresh product? On day 3, the transduced cells were then enumerated and then seeded in G-Rex platform, either 6M or 100M for expansion. At that point, everything is seeded level with different arms, fresh versus frozen. So same number of cells are seeded for each of them. And then on day 9, we harvested the cells for formulation and cryopreservation of quote-unquote final drug product. The wash was done on UF Mini with plasma-light HSA, and we added CS10 offline to that at 1-to-1 ratio. We filled in cryo cases and cell seal vials, as well as the standard 1 ml vials, as I mentioned, and cryopreserved in the CRF. Again, the disclaimer here is that the cryopreservation profile was an optimized, the cryopreservation process was an optimized step for the product. Now, looking deeper into the data from day 9 of the process, as I mentioned, the goal of the study was to understand whether the initial cryopreservation of the product has any impact on that final drug product, distribution of the cells, expansion yield, transduction efficiency. In all of the figures that you see in this slide, we have fresh versus frozen. So that's referring to the fresh leukopak versus frozen leukopak that went through the same process. You can see that by day 9, we have a slightly higher viability on the frozen arm and a slightly hit that the frozen arm takes on the fold expansion. Maybe it's about 10 to 15% lower than the fresh. Might be expected given that the cells were frozen, but there might be something more into it. We did look at the distribution of the cells and the composition of the cells that is coming in the next slide. Just so that this data is from donor 4, and the data from other donors are coming, and we certainly supplement this data and a slide deck with additional data that is coming from our lab. We look forward to future webinars and publications that will include all of those. This is what we have been able to generate at this point for this presentation, and we'll have more of that with additional data that is coming. Because we can't do every run and every donor at a larger scale, it's expensive for that matter. We wanted to establish comparability between the 6M and the 100M. So, to be able to do so, looking at the expansion yield based on the cells per square centimeter, which basically the third figure is the same as the second figure from the left. It's just expressed in the normalized fashion of cells per square centimeter. Excuse me. In terms of whether cryopreserved product takes a hit in terms of transduction efficiency. We can see that they are very similar. The cells that were frozen were transduced on par, very close to the same level to the fresh product. Through the great work of our scientist, Martin Campbell, in the lab, the target was, the transduction level was targeted to be at about 50%, and we are smack in the middle with 50% on the fresh product on day 9. And with frozen, it's just tiny, about maybe 1 or 2% lower with the frozen product, which is amazing work. Further characterization of that final product in terms of the co-stimulatory activation markers, looking at whether the cells are getting exhausted or not, looking at different phenotypes of CD27, CD28 expression, and also the phenotype of the cells in terms of naive versus central memory, effector and effector memory. In terms of co-stimulatory factors and activation domains, CD27, 28 are very much identical between fresh and frozen, with frozen group maybe just slightly higher on the double positive group of cells. One difference that we noticed between fresh and frozen is on the phenotype of the cells. One caveat is this is N of 1 donor, so we'll have additional data from different donors that are coming, and we'll see if that's a trend that that consistently shows up in other donors after expansion. But that's at this point, this is not a concern from what we generally know about the desirable portion of the cells of the cell product being the naive and central memory cells. We do have more naive population with the frozen arm compared to the fresh arm, which is interesting. It's not a negative score, I guess, on the frozen arm based on what we know, but that's a difference that we have noticed. So as I mentioned, we did this comparison all the way to day 9, characterize the cell product for the material that is generated from a frozen cell versus fresh arm. Now, at this point, what we continued on was we took the cells from the frozen arm and we continued that further into further characterization for formulation, cryopreservation, and post-thaw assessment of the final drug product. One goal that we had was to establish or understand the comparability between the various formats that may be used for that final drug product, be it in a research lab, if you're doing process development and R&D, and you don't have a lot of cells, you don't generally work in a larger scale, so it's not a closed process, it's frozen in the standard cryo vials. Or if you have a manufacturing process and you're thinking about using the cell seal platform, the small vials as the QC vials for the main dose that will be filled into cryo case. And we wanted to see how the cells respond when they are cryopreserved in these formats. Again, one disclaimer here is that we've done some assessment in our lab, some process development on the process of freezing, the thermal mass difference between these volumes, one being, cryo case being 30 times to 50 times larger than what goes into CSV2, CSV5, and standard cryo vials. And how does that affect the freezing process? We've done some optimization on that, and the results you're going to see is basically from an optimized cryopreservation process at that point. So we did look at viability and recovery of the cells post-thaw. And we also assessed the cells for the CD4, CD8, as well as other phenotype characterization. What we have on this slide is, you can see on the first figure on the left, the post-thaw viabilities, comparing that with the post-formulation. So on day 9, the harvested product was formulated in Cryostor and Plasmolite. For donor 4, it had about 90% viability at that point. Went through a post-thaw and then wash after thaw, and viability was regained to around 90% for the cryo vials and cell seal vials. The CryoCase platform shows a slightly lower viability compared to the smaller volumes. All the volumes show that delayed onset cell death in terms of drop in viability on day one when we put them back in culture. By the way, sorry I missed this. The way we assess the post-thaw is with an assay that we have developed in-house, the post-thaw outgrowth assay. Because these cells were generated in a G-Rex platform, after thaw and wash, we also seed them back in a G-Rex plate. It's a standard assay that we have developed. It's in a G-Rex 24. And we have done our assessment on optimizing the media volume, cell seeding density, and all that to make sure that the assay itself doesn't have an impact on the cell recoveries. So what you can see is that we are going to have some of that delayed onset cell death that the cells experience after thaw by that viability that drops to around 70% for all of these on day one. And then on day two post-thaw, the viability is recovering, and that is because the cells are back in business and doubling. You can see that from day one post-thaw to day two, we have population doubling of almost 100%. They're going up above around 105, 110% to around 200% on day two. So basically doubling from day one post-thaw to day two post-thaw, which is amazing. And one thing to note is how closely all of these formats, regardless of their volume, actually perform. So this is really nice. Basically confirms that the assessment and the optimization work that we did worked really well with this type of cell product. One thing that we note was the change in the cell phenotype or the cell type of the T cells. Again, through the great work of our scientist, Martin Campbell, this one was a cryopreserved product from a sequential selection starting material. So the target starting material was at a 1-to-1 CD4-CD8 ratio. By day 9, that is a smack in the middle. Again, that product at day 9 has a 1-to-1 CD4-CD8. But then upon thaw and then further culturing in the G-Rex platform, after thaw, we see a decline in CD4 population, which is interesting. We haven't seen a lot of reports of this change in phenotype, and that's something that we are going to continue investigating, and we'll have to look into whether this is cryopreservation related or it's an aspect of our post-thaw growth assay that we haven't really counted for. So this is an open investigation for us, and that would be something that we are very interested to understand if the cryopreservation has anything to do. Are the CD4 population more susceptible to the cryopreservation than the CD8s? Effectively, the ratio changes on that final product from 1-to-1 to about 1-to-2, maybe 1-to-3 by day 2 post-thaw. So that was some interesting observation. Obviously, this has nothing to do with the fresh starting material versus frozen starting material. This is the final drug product. It's a different conversation, but we thought we share this because it was very interesting. Again, as I mentioned, this was an optimized process, but what we demonstrated through this was we get a lot of questions, as I mentioned, whether cell seal vials and cryocases can be cryopreserved together, one as a QC and one as a final drug product, and this data is very much supporting that effort. And the last slide that I have is again on that final drug product. We did further assessment of the co-stimulatory CD4-CD8 domains. Aligned with that CD4-CD8 ratio change, we do see a shift in the CD27-28 presentation on the cell surface by 2 hours post-thaw back in culture. Essentially, we'll have more of single positive CD28 cells and less of double negatives, which is, I guess, is a good thing. and the single positive doesn't show up with CD27. So this is an open investigation for us on our end, see how does that match with that change in the CD4-CD8 ratio. Nonetheless, there was no impact on the transduction or GFP positive cells as we tested it in a GFP lentivirus. One thing we see is that the identical response from all the different formats and different volumes that we froze in this experiment, which is very encouraging. And then again, looking at the phenotype of the cells, the central memory portion of the starting material or the final drug product took a hit, dropping from 40 to around 15%, and the naive population increased. Again, we are working on understanding what it really means, and if there's a way we can eliminate that or reduce that by optimization of formulation or cryopreservation method. So at this point, I'd like to push this back to our collaborators at Carbiosystems and Bobby, and want to thank my team, Martin Campbell and Jesus Moreno, for generating this data set. We are still working to generate more, and I look forward to share more of this with you when complete.
Thanks, Alireza, for sharing that great data. It's always great to see the whole start to end of from the initial apheresis to the final formulation and harvest step. So in conclusion, as Alireza previously mentioned, throughout the last few weeks, Carbiosystems and BioLife Solutions have been able to really showcase the effectiveness of platelet washing from the initial Leukapack collection, going into either cryopreservation or straight into cell selection. From there, throughout this collaboration, we've been really able to showcase to you and other developers the ability to build reproducible, scalable, and flexible manufacturing workflows from starting material through final drug product, whether working with fresh Leukapacks or frozen ones and cryopreserved. So this data, supported by the ongoing studies that we've done thus far, really highlights that comparable performance supports the use of those cryopreserved workflows, particularly in that autologous cell therapy manufacturing space. So some key takeaways from today's presentation that we wanted to leave you all with was starting off with, ensuring that robust leukapheresis product processing. So ensuring we have everything from consistent viability maintenance to viable cell recovery, on top of ensuring we have the ability for automated washing, as well as doing it in that closed system processing from, that initial Leukapack washing stage. And then taking it into cryopreservation without compromising that quality. So we wanted to showcase and verify that we could achieve those really strong pre and post-thaw viable cell recoveries with very minimal impacts to viability throughout the process, as well as ensuring that we're collecting the right cells that we want to achieve at the very beginning stages of this process, which we were able to showcase today. And then also showing that downstream manufacturing performance, the ability to maintain that in comparison to other technologies. So having the effectiveness among cell selection, activation, transduction, as well as even expansion, really showed in comparison to other technologies as well that we can achieve these comparable CAR-T outcomes, as well as reproduce it, whether we're working with that fresh Leukapack or a cryopreserved Leukapack. So regardless of the workflow being done, we're able to maintain that performance, meeting all of these key metrics that you might be requiring for your workflows. Really just showcasing once again that, with the ability to cryopreserve these Leukapacks, we can have the automated, consistent performance throughout the cell therapy process, regardless if we're starting at the initial washing or going into the freezing and thawing stages. As Ali already mentioned on his slides, we also wanted to showcase on the CAR side, recognize the combined effort of both teams from Carbiosystems as well as BioLife Solutions. Alireza and I wouldn't be able to share any of this data today without the support of these individuals for a really strong collaboration. And with that, Alireza and I thank all of you for your time today. We're happy to take any additional questions.
Thank you both for the excellent presentation. We see a lot of great questions coming in. Thank you so much for that. Please keep submitting those via the Q&A box below me. We're going to address as many as we can, and the rest I'll pass along for follow-up offline. Our first question: for organizations currently relying on manual centrifugation, what is the biggest operational advantage they would notice moving to an automated closed process?
Yeah, I'll go ahead and I'll take this one. So really moving to that automated closed process, you have the big concern. One is going to be from a sterility and from a closed system process standpoint. So when we run an open manual centrifugation, there's that possibility of introducing contaminants. You also have the variability, as we kind of discussed earlier in the presentation, where depending on how the cells are being manipulated in the manual open centrifuge tubes, we have the concerns for consistent viable cell recoveries and maintaining the effective washing efficiencies needed for further downstream processing. Going to this in an automated closed system, you have the reproducibility with the recipe-driven automation and the ability to, have the cells be eluted and washed in the desired media output without having the ability to have to do any manual changes.
Thank you very much. You showed relatively consistent recoveries across different Leukopak donors. How much process optimization was required to achieve those results, or was the same workflow used across all donors?
Yeah, so I guess I'll take the first part of this one. So from the unifuge, the single-use centrifuge component of this, we really there wasn't much optimization that was needed. So the two key parameters utilized with the single-use tubular bowl centrifugation of the unifuge are G-force, centrifugal force, and flow rate. So it's really just trying to find that optimal set of flow rate and G-force to really maximize that viable cell recovery. So throughout the first set of runs that we had done, we had noted very clean supernatant lines when we were testing the waste stream to see if there was any cells being lost. We didn't really see any of that happening. So there really wasn't much optimization needed from the centrifugation piece of this.
And when you speak with cell therapy developers, what are the most common challenges they describe around formulation and cryopreservation?
I guess I'll take this one as well. So this one would be very much, at least from our side, from the centrifugation side, we tend to see some challenges associated with, from the formulation piece, having the inconsistent viable cell recoveries and just not being able to see if going in from the initial harvest to formulation, seeing the outcomes of the cells post-thawing to see whether or not we're having consistent performance. So at least for at least from the CAR perspective, we tend to see it quite a bit where it's, okay, going into the UF mini or the unifuge is metrics A, are we able to maintain the same parameters and metrics coming out of the unifuge? I don't know, Ali, Reza, if you have other insights on that.
Yeah, I guess, some of the questions that we get is around the processing time and the time that cells spend in that final formulated state, being hypoxic and hypothermic. Because typically the cell processing step takes longer in a manufacturing setting than in a process development lab, or when you're doing things manually very quickly. Things get longer because of all the verification steps that needs to go through, all the visual assessment that needs to be done, the batch records and operations that needs to be completed as things go. So things take longer. So most of the concern is around the timing and the stability of the cells. One of the major concern is the for use of BioLife Solutions buffers with Cryostore is the viscosity of this of the formulation and whether you can do wash or reformulation in any of these buffers. One thing that we were trying to do in this study to generate a little bit of data around, even if you don't do a wash in these, one thing that is possible to do is to elute the final product in either Cryostore base or CS5, and the use of CS5, or in this case CS10. So the good thing is, if you push out cells, elute cells in plasma light, you need to have at least a 1-to-1 dilution of that eluted cells with Cryostore offline to get to that 5% DMSO. And if you need to go higher percentage, depending on the cell type that you're trying to cryopreserve, that responds better to a higher concentration of DMSO, then you have to up that ratio to a higher value, and that dilutes the cell density. Now you have to freeze at larger volume. And one thing that we tested in this was we used the Cryostore base and also Cryostore CS5 in this case. to not only eliminate that manual step after reformulation or buffer exchange, but also have the ability to really adjust what that final concentration of DMSO would be at the lowest volume that you can get with the elution volume of the instrument in that higher volume up to 10% using CS10. In this case, we use CS5, but it's also possible to do that with CS10.
Thank you both. What additional data would you want to generate before declaring cryopreserved leukopaks functionally equivalent to fresh starting material?
I'll take this one. I guess additional data. So more donors. I mean, there's always a big variability from the donor material that is coming in, the quality of the material that is coming in, depending on how long it took between the collection and the start of the processing. And then there is also a bigger variability that whether the donor being a young person, an older person, female, male, and all the other demographic differences, as well as the state of the health of the donor or the patient at the time of collection. So obviously, we'll try to replicate this. This is, I think, fairly comprehensive. The only thing that we didn't really try in this one, and it really didn't pertain to the fresh versus frozen leukopak, was the functional assessment of the cells post-thaw on the final drug product. I mean, additional tests that we can do are cytotoxicity and killing assays, but that really doesn't speak about how the cells are generated to the point of harvest. I think looking at the assessment that we did in this study would be fairly comprehensive, looking at yields, looking at transduction efficiency, looking at phenotype shifts and changes by that and the end of expansion. It's fairly comprehensive, and we are working on generating this type of data from more donors.
All right, we have time for one more question. We have a multi-part question here. Do we need to get rid of cell clumps by filtration? What additional considerations to check the hold time? And are the analytical methods optimized for the cryoapheresis arm?
I can take this one. So cell clumps, with which part of the process, it's not clear what part of which material are you talking about. If you're talking about the AIF, and if the AIF coming clumpy, it's probably best to have a line filter before the cells are pushed into the bowl for washing. Obviously, you need to make sure that the cell wash equipment and the protocol is designed in such a way to effectively remove the platelets. As you know, platelets are activated upon exposure to cold and cryopreservation. If you have a large portion of platelets still remaining in that leukopak when it's formulated with Cryostor, when they come out of thaw, they will be activated and they can cause clumping. So I think you can eliminate a bunch of that issue upfront by upstream process development, and then you would have less of that clumping issue after thaw and continuation of the rest of the process. In terms of additional considerations for hold time, as I mentioned, so in this study, we had multiple parameters to look at. We focused on the variable or the variability from different donors. We focused on a couple of different wash methods. We focused on the wash buffers and elution buffers, as well as different formats of freezing the leukopak. And, so we had enough parameters essentially before we could add more parameters in here, but if we didn't want this to balloon up into this large, massive. As it is, it has a number of arms, and we haven't got into all of the arms that we've done. But we can certainly look into a stability of the leukopak before and after formulation. One thing that we did in this study was to compare the fresh material that was processed within 48 hours with the leukopak material that processed in less than 48 hours. That was to make sure that the hold times kind of represent a reasonable processing time on each end, each arm. In terms of analytical methods, that is optimized for cryoapheresis arm. Again, nothing would speak better to how the cells are cryopreserved and thawed than when you push them through the process of activation, transduction, and expansion, and you see identical results. That speaks for itself. So that's why we essentially performed this study this way.
All right, everyone, that is going to be all the time that we have left for today, but don't worry if we didn't get to your question. I will pass them along to our presenters to follow up offline. I'd like to thank Robert and Ali Reza for providing their expertise today, and you, the audience. We appreciate you being with us and hope to see you again soon. Have a wonderful rest of your day.